I'd like to just start by talking a little bit about some of the challenges when studying or modeling rare diseases and cancers. So some of the challenges are that we always have small sample sizes because they are rare. And then when we are looking at like multi-omic analyses, we're always dealing with the curse of dimensionality. So we maybe have a small sample size and then multiple features. And then it's also difficult to find external validation sets that we can compare our models to. And there's always heterogeneous data that, and if we're combining data from multiple sources or multiple institutions, we can have trouble with that. And then a lot of times we have missing data, and so we have sparse data sets, so that's also a challenge. And then there's also limited representation in public databases. So in cancer, there's limited representation in TCGA or COSMIC or things like that. Dr. Wong gave us a really nice introduction to explaining AI and machine learning models. But a lot of these techniques kind of create a black box, and it's especially true with multiple features. And unlike classical statistical models, the parameters and features don't have like beta coefficients or p-values that we can really explain to audiences kind of what's going on. And then when we're tuning our machine learning models, we don't have direct correlates to traditional statistical models. And so trying to explain what a hyperparameter is may cause some trouble as well. And then there's also complicated math that underlies optimization of AI models, and that can also cause some trouble. So two of the diseases I study, rare diseases, are mantle cell lymphoma, and it's a rare form of non-Hodgkin's lymphoma, and there are about 4,000 cases diagnosed in the United States. It can be aggressive or indolent, so the treatment kind of varies on the disease type. And then more of the aggressive type is subject to relapse and then disease progression after several cycles of treatment. And that was what I kind of focused on for this presentation, but my newest project is involving triple-negative breast cancer, which accounts for only about 15% of all breast cancers, and it lacks the hormone receptors that most targeted treatments are able to target. And it's also more aggressive, and it's found in younger women and often, more often in women of color. So this is kind of a visual abstract of my project in mantle cell lymphoma. We had 862 mantle cell lymphoma patients who were diagnosed at MD Anderson from the years 2014 to 2022. We extracted 129 predictive features from various modalities. We used a machine learning algorithm to accurately predict indolent versus aggressive disease. And with a pretty good degree, our AUC was 0.83. So this is just a broad overview. So when we were thinking about what model to choose, we wanted one that kind of fit with what we think about when we think about clinical decision making. And the way that clinicians make decisions is very much likened unto a decision tree. And so using something like an ensemble method where we are taking many, many decision trees and kind of combining them together was something we really wanted to think about using. And like Dr. Wong talked about, we used XGBoost with our data, and it was also very applicable to our data because we had a lot of missing data. And so this sparse data was suited to XGBoost, as XGBoost has kind of an inherent way of dealing with missing data, which was really useful for us. So one thing I talked about was explaining hyperparameters and how we tune models. And this is just kind of an overview of some of the hyperparameters that are tunable with XGBoost. For our project, we basically left open all of the hyperparameters except for the starting number of trees. And then we're able to tune that using basically a Latin hypercube grid search method. We curated data from, like I said, from different modalities. We had DNA data from targeted gene panels. We had it from whole exome sequencing as well for some of the patients. And then we also had some things coming from cytogenetics, which was kind of a broad view of the molecular happenings of these patients. So it was mostly from fluorescent in situ hybridization. And then we also extracted many features from the clinical record, including environmental exposures and just some kind of inherent features just coming from patients. So everything from BMI to different blood counts to reports coming from the pathologists. This is kind of our workflow, and I apologize for the kind of strange things on the slides. I did this in Keynote, and it didn't transfer so well to PowerPoint, but we started with pre-processing our data, removing zero variance factors, dummy encoding our categorical variables. We split our set into training and test sets, and then we also did 10-fold cross-validation for our hyperparameter tuning, and then of course chose the hyperparameter set that maximized our AUC, and then we fit our XGBoost model on the test set after we trained it, and evaluated the performance using different performance metrics. And then we also did some post-model analysis using variable importance features and Shapley additive values. And then after that, we versioned our model, and we launched it with a REST API just as proof of concept. So feature importance is an important part of making AI explainable. And we used two different methods. There are many methods out there. We use Shapley values, which was discussed before, and also variable importance plots, which are a permutation-based approach, and it could be either model-specific or model-agnostic. And for XGBoost, it uses information gain, or basically the fractional contribution to classification based on the splits in the trees. And these are two examples from other models, not ours. This is from our model. When we fit the model on our validation set or our crossfolds while we were training our model, we pulled out the top 20 important features using variable importance. And they range from all of our different modalities. So we have things from somatic gene mutations, different measures from blood counts, morphology from pathology counts, and different things like that. And we took these 20 features and basically took our 129 features, and we wanted to make a parsimonious model that was more clinically applicable. So we took these 20 features and we built a whole new model that would be more useful in clinical practice. We then tested a bunch of our different models. So we had our all features models, which had 129 features. We had an only clinical model where we separated out the clinical features and built a model using that. And then, of course, just next generation sequencing, we did cytogenetic only, and then our parsimonious model that was only 20 features, we compared with that as well, and you can kind of see the different metrics there. And the parsimonious model performed almost as well as the all features model, which was an important thing for us to demonstrate. And A and B are the Shapley values and the variable importance plot from our all features model. And then C and D are from our parsimonious model. There are many overlapping features from the two models, but it was very informative to us, and especially our clinical collaborators, to see that a lot of these important features were reflected in prior published research. And so we did find a couple of surprising things. And one thing we found is that some of the cytogenetic features weren't as important as previously thought, maybe, and that these gene mutations were kind of factoring more into this. And it actually wasn't a surprise to the pathologist we worked with, as he said that looking at the DNA gave us a more granular view of what was happening on the molecular level, and that cytogenetics were really kind of a generalized or broader view. So that was kind of interesting. We also took those 20 features, and we wanted to show that this could be a viable feature selection method for classical statistical models. And so we utilized that in a generalized linear model and also in survival models, because we knew that our problem was a little bit of a censored regression problem as well. We were distinguishing at baseline who we thought were going to be the patients who had aggressive disease versus the patients who had more mild or indolent disease. And this really kind of played into, something that was more applicable to survival models. And so we fit those 20 features into Cox regression models and for progression-free survival and overall survival, and then also did some univariate log rank tests just to kind of show that by picking out these features, we could kind of do some more analysis to really show kind of what the prognosticability of these features was. And this is just a table from our publication showing the different performance metrics of these models. And like I said before, our all features models that had 129 features, our parsimonious model with only 20 really performed just about as well, which was very interesting to us. So just briefly, because I know we all want to go to lunch, but with triple negative breast cancer, we are conducting kind of multi-omic studies on a cohort of patients enrolled in what's called the Artemis trial, and these patients are receiving various targeted treatments. And for these patients, most of them have both genomic and gene expression data. We also have radiomics. We have some digital pathology data, and we're hoping to kind of integrate this in the future into a larger prognostic model. And so one example that I thought of that we've used so far, where we're explaining kind of a biological feature from many, many, many different features or data points, is looking at mutational signatures. And so there are thousands of somatic variants from the patient's tumors, and we wanted to know what variants are important in the mutagenesis of cancer. And so can we extract these molecular characteristics or signatures in our cohort? We use non-negative matrix factorization to compare base substitutions in genes to the existing databases of matching signatures. And this is actually from our cohort, and we were able to find 4 different signatures that kind of related to previously published tumors, and it does give us some kind of insight to what's happening biologically. The exposure to tobacco was interesting to us, not because we think these patients were exposed to chewing tobacco, but because this might represent an exogenous or an environmental exposure as well. So in summary, explainable AI and ML models have broad application in precision oncology and studies in rare cancers. And these algorithms can be used to select features for further machine learning or classical statistical modeling and analysis. And many model-specific and agnostic approaches to AI exist. There are always strengths and limitations with using these methods, and we should never neglect biological intuition or interpretability, and also keep in mind the practical applications of the models, and can you use these in clinical practice. So I know that was kind of fast, but I did want to acknowledge especially the patients and their families and their contributions to our research, and all of the folks on my research team, and all the sponsoring institutions, and my funding and support provided by the NCI. And there's a link to GitHub for the model code that we used in our MCL project and our paper that was published in Cancer Research Communications. So thank you so much.
Yeah, that was really interesting because I also work in explainability. And so my question is, there is one thing called SHAP. So what your viewpoint if we want to build our own explainability anything? So what should be the approach? Should we go from the hardware perspective, or should we go from the mathematical perspective? Because the SAP is completely based on game theory. So what will be your approach?
Yeah, so I think it's important actually to look at many different approaches and really keeping in mind what question you want to answer. But I like your thought about, Shapley kind of using that game theory where it's like all these contributions, and it's very model agnostic, right? And then looking at, other methods where we can identify the important features. So even using, classical statistical models where we even test it out on the same data set and see kind of what happens. So.
And thank you so much. The second question is, you did the feature ranking based on the SAP values. Have you tried to do the SAPly interaction values, and did it changes anything, any changes in the feature ranking?
Yeah, so that's a great question as well. So interaction is something we thought a lot about. And we actually did look a little bit to see if we could see any interactions between the features, and we didn't with our kind of, I guess, limited data set. But I think especially with multi-omic data, there's a lot of potential for that. Kind of looking at those interactions.
Yeah, because I tried with the blood pressure data, and I see a clear difference between the SAP and the SAPly interaction values.
Yeah.
Thank you so much. The last question. So this is all about the tabular data. So have you ever thought of any literature regarding to the image data? Because a lot of people work in the image data sets, and what explainability we can bring in the image data sets, because there are a lot of different types of images, histopathological, confocal, X-rays, radiographs. So how we can bring explainability to those models trained on image data sets?
Yeah, integrating different modalities is something we're thinking a lot about, especially with this breast cancer project, as we do have a lot of like radiomics, and we also have digital pathology. So being able to, I think, pull out the features or make composite features from these different modes, I think would be really important, especially in explaining our model, right?
Thank you so much.
Thank you.